me here. So my name is Wennan Chang. I'm a 5-year PhD student. And actually in this summer, I'm in an internship program in the Kite Pharmacy. It's a drug company. It's located in the Santa Monica, California. But due to the COVID-19, so the office is closed. So actually I'm working in a remote mode. So I work from my home. Yes. First, yes, I think we can start.
Oh yeah, and within this mechanism, so Pengtao will handle the, I think Pengtao is here, Pengtao will handle the questions, so you don't need to be bothered a lot by the question in the chat, but whenever you see it, you want to answer it, so you can stop and answer, but if not, you can just leave it for kind of adding the end.
Okay.
Yeah.
Okay, thanks. Okay. So before I start my topic, deconvolution, so I want to share some my internship experience. So my company is called Kite Pharmacy. So actually this company focuses on develop some drug or produce some related product to cure this diffused large B-cell lymphoma, this DLBCL. So this is the one horrible type of cancer. I just choose some image here. And so if those B cells cannot be controlled by our immune system, so it becomes a malignant cell. And those malignant cells will diffuse different part of in the body, and it will accumulate, become tumors. So what this company do is they use this engineering the T cell therapy. So this is till now it is the most advanced the treatment for these cancers. So what they need to do is the patient first need to go to the local hospital, which has a collaboration with this company, and they need to collect the patient white blood cell. And then those samples will ship it back to the lab, to their company's lab, or maybe to their nearby labs. And their staff will do the isolation, will isolate the active T cells, and then they will add it to some. Let me check that. And then they will add it to the gene of those T cells to make them have the new functionality, and then they produce or copy a lot of this new T cell. And then they will ship those samples back to the local hospital and infuse into the patient. So this is amazing progress. So I actually during my orientation, so I have been show some videos. So how the patients looks like before the treatment and how this patient looks like after the treatment. So this is amazing since maybe part of the patient will have very great outcomes. So those malignant cells, those symptoms will just disappear. Oh, it's very fancy for me. So what I want to talk here is so actually what every day maybe we do in our lab is not that far away from the real world practice, real world case. So maybe what we do maybe can help the doctors in the hospital. Maybe we can maybe what we do can help the patient. So before I start my deconvolution topic, so I will give some introduce what's the cancer or what the tumor microenvironment looks like, and what is the cancer immunotherapy. Yeah, let me share this video.
Many genetic and environmental factors can cause tumours to form. For instance, UV radiation from the sun can damage DNA and other structures in melanocytes, which are the pigment-producing cells in the skin. Chronic damage to melanocytes by UV radiation leads to most cases of melanoma, which is a type of skin cancer. As melanomas grow, they can eventually spread to other sites in the body, such as the lungs and liver. The cells of the immune system are continuously monitoring our tissues. Natural killer cells, or NK cells, sense stress-associated molecules on damaged and cancerous cells. Dendritic cells, or DCs, activate cytotoxic T cells, which can then sense tumor-associated antigens using their T cell receptor and other co-receptors. Once activated, NK cells and cytotoxic T cells release perforin and granzymes. These molecules punch holes in the surface of the tumor cells, causing them to die by apoptosis. Helper T cells support these responses. They help DCs to activate cytotoxic T cells, and they produce cytokines such as interferon gamma that recruit and activate more NK cells. As the tumor evolves, genetic changes occur that can give some tumor cells a survival advantage. This means tumors are often heterogeneous. For instance, tumor cells may no longer express the molecules that are sensed by the killer immune cells. As the immune system continues to kill the tumor cells it can recognize, the cells it can't sense become more prevalent. This is immunoediting. It leads to the emergence of a tumor that can't be detected by the immune system. Some tumor cells actively suppress T cells by expressing inhibitory molecules such as PD-L1. PD-L1 binds to the PD-1 receptor on T cells and deactivates them. This is an immune checkpoint. In addition, tumor cells can attract immune cells that suppress the activity of other immune cells, thereby supporting tumor growth. These immunosuppressive cells include regulatory T cells and certain types of myeloid cells. Therefore, the tumor microenvironment is like the scene of a battle between two opposing immune responses. One side of the immune system is attacking the tumor, while the other side is helping it to grow. Scientists are developing immunotherapies to help strengthen the immune attack. One approach is adoptive T cell transfer. Cytotoxic T cells taken from patients with melanoma are selected for their ability to attack tumor cells. The best killers are infused back into the patient. Treating patients with cytokines, such as interleukin-2 and interferon alpha, can also boost the activity of anti-tumor immune cells. An alternative approach is to target the immune checkpoints. For instance, antibodies that bind to PD-1 stop this molecule from switching off cytotoxic T cells. Another immune checkpoint being targeted is CTLA-4. Blocking this molecule helps DCs to drive anti-tumor T cell responses. Not all patients will respond to these immunotherapies, and some responses will be delayed. Combining immunotherapies with chemotherapy or radiotherapy can lead to a better response in some patients. Immunotherapies can themselves be combined. For example, PD-1 and CTLA-4 blockade can improve responses when administered in combination. Activating the immune system has risks. Some patients develop harmful side effects when their immune system attacks healthy cells. Nevertheless, there have been encouraging results from clinical trials. This animation has focused on melanoma, but it's becoming clear that immunotherapies can be used to treat many different types of cancer.
Yes, to me, that's a good video. I think it's very clearly to introduce what the tumor microenvironment looks like, and how the different immune cell types works, how they're doing their jobs. And in the 2080, so the American James and the Japanese scientist Tasuku, they won the Nobel Prize of the medicine for their discovery of the cancer therapy by inhibition of the negative immune regulations. And this American scientist, James, he is the researcher of the MD Anderson Cancer Center located in the Houston. So he discovered this protein called the CTLA-4. So I believe all of you already see those keywords like the PD-1 and the CTLA-4 in our previous videos. So and this Japanese scientist, he discovered this PD-1 this protein. So those two proteins is very important in our treatment, in our current stage of treatment. So actually, this treatment is very promising and very amazing. So since we have know some background about the tumor microenvironment, and maybe I just give you the definition. What is I just give you a definition. What is deconvolution? So actually, what is deconvolution? It is just a computational technique. The goal here is to estimate the proportion of different cell types in sample collected from a tissue. So why the deconvolution is so important? Since it can give our intercept of what the TME looks like for different patients. So this deconvolution will give us the hint what the TME looks like for different patients, and this can give us some advice for the doctor. So how can we treat this patient? So maybe we should use different strategy to treat the different patient. And the deconvolution here, the goal is we want to know what it looks like in different tissues. For example, the doctor maybe have the tissue taken from one patient, and then they will use this tissue to do the sequencing. I believe you already been introduced the RNA bulk data, and maybe just like the single cell data. So here, the tissue here means we use the bulk data. So it's just the mixture signals. After I get those data, so we will use our method, or we will use some other method to get the result. So I will feedback to the doctor. So in your specific microenvironment, so maybe how much immune cell type there is, and maybe I will also feedback for each cell type what is the related cell type proportion, or not, or maybe it's what is the cell type fraction for each cell type. Maybe for the B cell, you have maybe 20% of the B cell, and maybe you have the 30% of the T cell, and maybe different T cell, CD4 T cell and CD8 T cell in different resolutions. So this is deconvolution. It might be still confused. So I try to find some similar problem. What is a deconvolution? So in the computer science field, there is an old problem called source separations. So here in the class, in our music class, we can record some signals. Here we call it the mixture signals. These mixture signals may be combined with some source from instrument and some from the human sounds. But all we have is we have these mixture signals. So what we wish to do is we wish to use a computer to separate those source and to estimate the original source. So this is also a very difficult problem. But what we have is we should depend on some prior knowledge. In this case, the prior knowledge here is the frequencies. For example, the piano and trump and the violin, for different kind of the instrument, we have different frequency. So even for the human sounds and even for the dog bark, so it's totally different in the frequency domain. So how we do is we use this prior knowledge to separate the mixture signal, and the output is to estimate the original source. So this is just like the deconvolution. So deconvolution, what we have is a mixture gene expression file from the patient. What we want to do is we want to tell the doctor, we want to tell the patient, in your specific micro environment, so what immune cell type do you have, and what is the related cell type proportion? So this is very important. So even we can predict the outcome, or even we can give advice, the specific called precision treatment. So in this source separation, we have the prior knowledge called the frequency. But what the prior knowledge here we wish to use to do the deconvolution. The answer is we use cell type marker genes. Since the different immune cells have their own functionality in our immune systems. For example, some cells will just recognize the virus, like the COVID-19 virus, and just transfer this signal to other immune cells. And some cells or some immune cells, they have functionality. Maybe they will to generate some antibody. That's how our vaccine works. And other cells, maybe like NK cell, they will kill the virus, and maybe they will kill the diseased cells, like the cancer cell or malignant cells. So how they do their own job is because they can produce the different proteins. Those proteins really depend on the different genes. So for those genes, I call those marker genes. And what we do here is we collect purified immune cell. For example, we have the purified cell line. We have the B cell line, T cell line, macrophage, and maybe we have the NK cell lines. And we collect them for each group. And then we will do the sequencing for each group. Those how the gene expression profile looks like for each cell type. So since they have the different role in our immune system, so actually they have a totally different gene expression profile. Here we have a specific terminology. So if we combine those information, we call this signature matrix. Matrix is just like the large table. We can also call this a signature table here. So we wish to use this as our priori knowledge to do our job, to predict our cell types. But for the user, our user may be just a normal patient or normal doctors in the hospital. Actually, they don't need to care about what's the priori knowledge we use or how our complex algorithm works. So what they need to care is just the input and output. The doctor just takes the tissue from the patient, and then they send those tissues maybe into our core lab. So we do the sequencing. We have our technician staff to do the sequence things. And those staff will transfer the data to me, and I will try our best to use different methods to predict the output. The output is in this specific tumor microenvironment. So what immune cell do they have, and the related cell type, and the related cell proportion for each immune cell type. So this is what they need to care about, just the input and output. And I think in my topic here, I will ignore the most complex algorithm part, and I will just introduce this important thing like the input and output, and also the definition of the deconvolutions. So I think we still have the 40 minutes, and I will introduce 3 methods. For each method, maybe I will use 10 minutes, and I will give the demonstration. And if we have time, I think we have some code. We can run that, and if we have questions, we can discuss and solve that. And also, I wish you can maybe try the servers, try our servers and try different servers. And I think it's very helpful to let you understand this topic. The first method I want to introduce is called the SADC. So this method, they use a single cell as a prior knowledge to generate the signature matrix. So let me open my RStudio. So I think you already familiar with this interface. This is just the programming interface. Before I start run I just read this demo code last night, and this let me remind my first class in my bachelor degree how my computer science teacher introduced the code to us. And the most important things I think if someone wish to become a programmer or coders, I think the most important thing is to write a clear and the comprehensive annotations, maybe for the documentations. For some companies, they will require the comprehensive and the detailed documents for each programmer, for each code. So this will give maybe, for example, if your other colleague will use your code, so they can first read your documentation and then you use your code. So I just read this annotation last night. The first, I will begin our code. So the first, we need to change our work directory to our folders. Since in our folders, actually, I prepare some data sets. I will introduce this later. So here I use a Windows system. So I just set my folders here. So if you use Linux or if you use Mac, you just give new folders and set up here. And if I want to use this package, I need to install this package first. But here, I already installed last night. So if you, I think in this step, you maybe face some error or face some problem. So please feel free to give Pengtao an email and maybe to me an email. So if you have error. After you install those specific package, you need to load those package into our workspace, into our current workspace. Yeah, let me do these things. And yes, and this package, this software, they give a comprehensive websites. So what this package can do, how they works, and also they give a very detailed examples. So I just choose one simple example here. I just want to show a simple demo, and they also have the pre-processed data sets. For example, those single cells data sets, and this is the bulk tissue data we wish to use. So this is from the human pancreas accident data sets. So actually, this is not from the tumor, but is from just the normal tissues. But in this normal tissue, we have maybe 6. I think we have 6 cell types. So if we have prior knowledge information, and we can use this prior information to predict what's the cell type proportion in this bulk tissue data. And they also have some other data sets, but I just downloaded those parts. And let me show my folder. Yes, I just downloaded these things, and we need to use this line to read this data into our workspace. This environment is to show what variable we have in our current workspace. Let me run this lines. Yes, we already have this one. So after we load this variable, I always do is first I want to check the class of this variable. Since the different package will use different object. So for this case here, this variable is a bio-based class. So since we can also click this variable to see how the data storage in this variable. And then I want to see what's the big table or what's the mixture gene expression file looks like. It's a large matrix. And I use this command to show, oh, it actually has the 32,000 and genes, and they have the 77 patients. So this is the size, or this is the dimensional of our bulk matrix. And this is not so vivid, so I just want to do some visualization for this bulk RNA-seq data. So I need, if I want to use this heatmap tool, I need to load this gplot package. First, load these things, and then I use this function to draw the heat map for our bulk data. Yeah, I just only show the part of the data, but this is what our data looks like. This is our bulk tissue data. So for each column, each is a patient ID, and for each row is our genes. Here the number means gene expression value. So this is our input. This is the this is the material the sequence staff transferred to me. And then we will load some priori information. Here I use the same code to run this to load. We can see that it is already in our workspace. And I want to show some of the single cell statistic of these data sets. And I will introduce this plot for detail. Let me maybe zoom. Yes, in this priori knowledge, we collect maybe I forget, maybe more than 10,000 single cells. But in total, those 10,000 single cells from the 6 patients, here H means health. You can find all this single cell collected from the health patient. And this is a single cell number for each patient. So and this bar plot also show the detail for each cell type. We have the exactly how much single cell there, and this is we scale this each bar into a 100%, so we can know the percentage here. So we can find the alpha cell have the large percentage in our priori informations. And then I want to use their main function to do the deconvolution on the human pancreas tissue bulk data. Let's do these things. The first parameter input here is our bulk data. The second parameter here is we want to input our priori information, and then is some other parameter. We can select our interest cell types. In our pancreas Iceland datasets, we have these 6 cell types. We have alpha, beta, delta, gamma, ethna, ductal cell types. And let us run this major function. Since this deconvolution is computational heavy, it should take some time. So till now, does anyone have some questions? So maybe, oh, let me check my chat. Oh, yes, this SADC is available to everyone. So I think it's totally free. They have maybe academic license for use. So let me share the calculation part is already done. But let me share this link to our chat so everyone can touch those materials. Cool. Yes, so our major computation is done. So after we get this result variable here, so we want to check the output format. So I will use this dim function again to check the, it have the 15 rows and 16 cell types. Since for this bulk data, we have total 77 patients, but I only want to calculate the normal patient. It means another patient, another group is like the diseased patient, but I just want to calculate from the normal group. So in our normal group, we have 51 patients, and we have the 6 cell type. Let's print only the first 6 rows. Use these head functions. And click run. Yes, this is the patient ID, and this is cell type names, and this is a cell type proportion. So for example, for the patient 1, they have this 0.45%. They have this 45% of the alpha cell, and also they have a lot of the ductal cell in these tissues. And but this is sometimes we will just transfer this, and we will just save this result as a CSV table and transfer back to the doctors. But sometimes we will also give some plot. For example, we can do a heat map for these big tables. This is our predict cell type table looks like. So each row here is each patient, and each column here is cell types. So the red means the larger percentage, the blue one means the small fewer percentage. And if the doctor interested to one specific patient, we can also give them a bar plot. Here I need to load this color package. For example, if the doctor interested in this patient ID 13th, I will just replace 13th, and I will draw the bar plot. Or we can have a clear information what's the tumor microenvironment looks like for this tissue data. And if we interested into patient number 15, so we can change this variable and we can do the bar plot again. So we can find their microenvironment is different. And we can also try this one, patient number 5, and do bar plot again. Yes. So this is all the demo for this first method SADC, and but here I just write a lot of the annotations. And yes. And the second method I want to introduce is our in-house developed method called ICTD. Since I know even for you, it's difficult to run the code because you need to install the R software on your laptop, and and sometimes you need to download the different package. And this is a very frustrating posture. So we wish to make everyone happy. We wish to make an easy life. So we build these servers, and the background is similar code, but we just give this simple server to let the doctor can use this very easily. So this is a major interface. Let me show. Yes, this is our website. The server is hosted in IU. So we have let me introduce a different tab. For the About tab, we have the detail how this algorithm works and what's the detail for each step in our algorithms, and also some abstract and some contact information if the doctor has the problems. And we also have some part like FAQ, and we also have some other software in our labs. And I think the most important thing is we have this clear tutorial. So I will show you this one. And the first one, what's the doctor to do, or what's the user to do, is to click this browse button to upload their data. So actually, in our web server, we have a default data. So if we leave this black, it will run our default data sets. And the user also need to choose what this tissue come from. It's from human cancer, or it's from the human brain, or it's from the mouse cancer. Since in the IU school, so we for some lab, they will discover the new mechanisms of the cancer. They will use a mouse to do the experiment. So they will have a lot of data sets from the mouse. So the mouse and the human, they have a little bit variation. So we build a specific priori information for the mouse. And then, since we collect our priori information before, if you are interested, you can download our cell type marker gene here with these 3 links. You can download our priori knowledge here. And that's all. All you need to do is done. You just need to click this button, run ICTD, and wait. Since this is our Shiny servers, it will take several minutes. So I just will go to the first page. I just leave this blank to run the default, or to run our background data sets. and I just use the default parameter. And you can use this link to download our priority information and click run ICTD. And after the run, we can get the result. Here we can get the table just similar to our coding parts. So this is a table, but I just only show parts results. I just show the first 6 patients. For each patient, you can have the cell type, predicted cell type, and you can also have the cell type proportion for each patient. And I also plot a heat map for the overall table, for the overall results. So you can find totally we have the 30 patients, and for each patient you have the immune cell type, and you have the color represents the cell type proportion or cell type fraction. And after we get this result, you can download this table by this link, save as a CSV file. So you can use this. To generate your own plot here. And also, since our method can give the data-specific top marker genes for each cell type, you can change the tab here, and you can use this link to download those information. Yes, you can download this. You can download this cell type marker genes. And if you are more interested for maybe one specific patient, you can here input target sample ID, and this bar plot will update instantly. So let me check whether, oh, it's still running. But after this, after we get the result from here, I will show you. Let me check whether we have questions. Oh, thank you so much for sharing this link. But I think our server is kind of crowded. So if a lot of users, maybe it will very lazy. But we can wait a little bit. Yes, finally, the computational procedure is done. Yeah, we get our result here. We just show the 5 sample here, and we can see the B cell, T cell, fibroblast, endothelial NK cell, and this is related cell type proportion. And here, this is a heat map. And the default sample ID, I just random give the number 3 ID. The patient number 3 is this one. But maybe I think there are maybe I am interested for the patient number 1, so I just maybe input number 1, and yes, this bar plot will update automatically. This will give you very simple and very clear information about what the microenvironment looks like. And maybe if we are interested in this patient number 27, and click the enter, yes, it will draw this bar plot instantly. So, and we can download this predicted table to here. It will give you a default name. Let's check. And we also have this marker genes. So let's also download this marker genes as a CSV file. And then let us check those results. Yeah, this is ICTD cell type proportions. Yes, this is our result. And let's check this marker gene predicted by our method. Yes, this is a cell type marker gene predicted by our method. So that's it. This is our own methods. So actually, the Professor Zhang and I, we developed this method together. The final method I want to introduce is called Cybersoft. This method is developed by the Stanford University. First, let me copy this. Let me copy this page to our chat. This web server is really fast, but it required you to register a card and to use your email and create a password. I already have my own account, so yes, maybe since the time is long, so I already be logged out, but I will use my account to log in. So this is a pipeline of this method, how this method works. They have their prior information, and you can upload your own data, and then you will get the output here. Let's go to the About page. Yeah, this is about some detail about their funding and implementation and the overviews. And there is also some contact information and who maintain their servers. If you have questions, you can write email to them. And this is a major part. I will show you this now. So in the menu, we have the run Cybersoft, but we can also have the tutorial and FAQ and maybe history version. And we can also upload the file. Since I never upload, since I upload a lot of things onto their servers, we can try those different data sets I collect. All these things are from the TCGA database. It's from the cancer. It's different cancer types. And the job result, I will go to this one to run their method Cybersoft. Let's see. So first, we need to choose a signature gene file. I think you remember this signature is our prior information. Actually, in their method, they collect this prior information named LM22. It means they have 22 immune cell type. They can give a very high resolution for the microenvironment. And then we should choose our input file. This input file is our tissue bulk data. So since I upload a lot of things, maybe I will choose one cancer type. Maybe I will choose this one. I will choose this SKCM. I think it's a melanoma cancer. So this is the other parameter. Maybe I just choose a permutation 50 times. And the custom gene sets. No, I don't have the custom signature genes. And I don't want to use their example data. I want to use my own uploaded data. And then I click run. It will take some time. Since the server is running, so if anyone has questions, you can just type your question in the chat and we can discuss. Maybe I will let it run and I will show you my previous result. I think it will save us a lot of times. Oh, there is a question. I noticed on the Shining sites that there was also brain data available. That part. Here for the human brain, so we use our collect data to train the human brain signature matrix and their own markers. Is this part of the brain? No, I don't think this is a part of the Brain Institute and so the NIH. Oh, yeah, we have a question about the funding. Yes, I think, for example, for this Cybersoft, if you are an academic user, if you are working in university or in the hospital, so actually you can use this software for free. But if you are a commercial, if you are a company, for example, in my summer intern, I also implement some deconvolution method for that company, for that drug company. So actually, they cannot use Cybersoft because Cybersoft only gives the free for the academic user. So if you are a commercial user, you need to write them email and you need to talk about the price. Yes, but for our Chinese, but for other methods, actually, that's how I think that's how the academic works. So actually, NIH or NCI, they will give the funding to generate this advanced method and also to generate this user-friendly servers to help the doctor in the hospitals. I think that's the mechanism for the academic. So if I'm wrong, maybe the Professor Zhang can correct me, since I'm not familiar with the funding and all about money things. I just need to focus about the developing my methods.
Yeah, but for sure. So yeah, so I think your point is right. So for sure, so for the industrial applications, so for this kind of for this academic kind of a work, so academic product, sometimes they may need to definitely need some license, but since most of the softwares what they're based on is not kind of very hard, very complicated algorithm or computational approach, or actually more importantly, it's kind of it's just a computational concept. So I believe for most of the companies, they have their capability to implement those methods by themselves, and also with changing some like the core methods by using like definitely like something like there are some substitutable approaches. So I think for this type of methods, it won't be a big issue. Yeah.
Yeah, thank you so much for the explanation. So since this running is still in progress, so I just show my previous result. So I click one my previous job, so I can find this heat map table. So actually they have 22 immune cell types, and they have multiple patient ID. For each patient ID, you can see the related cell type proportion for each immune cell type. And also you can download this table in different format. For example, you want to download it as a PDF, or you want to download as a CSV or TXT file, it's up to you. Yeah, this is about this method. And that's all. I have done my total, all my materials of this topic. So thank you so much for your attention.
Thank you.
Do you see a lot of use from the IU labs on the deconvolution software that are the tool that you guys have developed at the IU server? Do you see much use on it from labs around IU?
Actually, we are still submitting this